Getting very focused. Very focused. The front of the room. What's another name for non-specific defense or non-specific immunity?
General.
You can try.
Innate.
I heard innate. Yes, it's innate. So non-specific defense, this is what we're going to finish today, and then immune 2 is all about specific or adaptive immunity. So this is a slide that we had to finish over considering inflammation. So here you are. You're working in the lab. You cut yourself on a beaker. So you've injured your tissue. And this flow chart is going to show you what happens when you do injure your tissue in terms of inflammation. So here's the initial stimulus in pink, tissue injury. And then, I don't know if you can really see these, it's kind of a, what color? It's like a greenish, I don't know. What kind of green is that?
Colorblind.
Oh, you are? Well, I'll point out to you then. Which ones are words. There's the physiological response, and then in red it says signs of inflammation. And then orange down at the bottom will be the result of inflammation. So, you injure your tissue and you release these things called inflammatory chemicals. We already know this one, histamine. Which cell type releases the histamine? Basophil. Yep. Pardon me?
Always have my friend the basophil.
That's so dorky. I love it now. When I go to the nerd convention, they're going to give me that. Sure. My friend Basophil. Okay.
What did she say?
She says, I'll call my friend Basophil. Nerd convention. So, anyway, you release, in addition to histamine, there's these other inflammatory chemicals. And it shows over on this side that you can also release this thing called leukocytosis inducing factor. And then if your body, in response to inflammation, undergoes leukocytosis, that means you're just increasing the number of white blood cells. Where would leukocytosis inducing factor, where would that molecule have to go to make more white blood cells? What part of the body?
Red bone marrow.
Red bone marrow.
Red bone marrow. Yeah, good. Perfect. So we kind of already talked about this in the cartoon I showed with the splinter going into the skin. But what do the inflammatory chemicals do? Why do you want the tissue that got injured? Why do you want it to release inflammatory chemicals?
You want it to try to like separate from healthy tissue? Maybe, I don't know. Okay, maybe not.
Well, you want to protect the healthy tissue from the damaged tissue, so that's kind of a seperation.
Kinda.
Yeah, that makes sense. I see what you're saying.
Does it?
Oh, go ahead. All right. I am. Just kidding. All right. So, what you want to do, the area is damaged, the tissue's damaged, bacteria is in there, most likely. You want your blood vessels, in this case, arterioles. Arterioles are really small blood vessels. You want them to vasodilate. And we talked about this last week. Why do you want the blood vessels, arterioles, in the area that got damaged? Why do you want them to get bigger?
To attract more of the right molecules to the area to do the job.
Yeah, so if you have a larger volume, you're bringing more white blood cells to the battlefield, to where the damage is. So the arterioles dilate, bring more white blood cells to the area. And why do you want your capillaries, they're blood vessels again, arterioles and capillaries are blood vessels. Why do you want the capillaries to become more permeable? And use a cool sciencey word to explain why you want that.
Follow the concentration gradient.
Okay, you're on the right track, but one step before that.
Margination.
Yeah.
One step after that.
After that. Diapedesis.
Diapedesis, that's when the white blood cells crawl through the capillary wall. if the capillary wall is not very permeable, then the white blood cells can't crawl through it. So you want, between the endothelial cells of the capillary, what are those structures called that you want to become leaky instead of tight?
The junction?
Yeah, tight junction. Way to jump on that. So the tight junctions between the cells in the capillary, the tight junctions loosen, and now there's big space between the cells and the endothelial cells, and then the white blood cells can crawl through, get to the battlefield. I should show you that picture, because I'm up here just chatting away, and I have this beautiful picture in my mind, and I'm hoping that you all have it in too. This is what is in my mind right now. This is an endothelial cell, right? They line the blood vessels. We just had that on the exam, so everybody knows that. Endothelial cells become leaky, increases their permeability, and then the white blood cells can crawl through to get to the battlefield. That's the cartoon I have in my head. And now you have it in your head. I really like it. Okay. And then it says here, this is all, remember, this is all part of the inflammatory response. All of these things, you've all had inflammation. All of these things happen when you have inflammation. And then you said, Jess, you said chemotaxis. So that's when the neutrophils and monocytes, they'll crawl along a chemical gradient to get to where the bacteria are. But in order for these three things to happen, you have to have release of inflammatory chemicals from the damaged tissue. When the arterioles dilate, you get a local, wherever the arterioles are. So in this case, the local hyperemia would be in this area where you got the cut. And we said arterioles dilate, that's going to increase the blood volume and the blood flow to the area. Increased capillary permeability is going to lead to fluid leaking out of the capillaries, and then that area around the finger will become, what's the cool sciencey word for that when you get leakage out of the blood vessels into the tissue? Starts with an E. Edema. Yeah. And you've all had that in inflammation, right? Wherever you get cut, it gets a little swollen. Okay. And then, oh, wow. Man. I have to get a humidifier. It's so dry. My throat is killing me. Does anybody use a humidifier?
Yeah, it's awesome.
Is it good? It does?
Yeah.
Okay, yeah. I gotta get one of those. So, here, who said margination earlier? Somebody talked, yeah. So here it is in the flow chart. The chemicals will add, those inflammatory chemicals will tell the leukocytes to marginate or cling to the capillary walls. And then what happens is when the arterioles get bigger and more blood is flowing through, that area is going to get hot. And you know that, right? And it's also going to get red because if you have a larger blood vessel bringing more red blood cells through, it's going to look red at the skin. Swelling, that's edema. And with all this inflammation comes pain. Why do you have pain? Well, histamine may be a molecule for pain, but what I'm thinking is, remember from 201 when we looked at the skin, the integument, and there were pain receptors in the dermis and epidermis? Those pain receptors just got cut, so they're going to be throbbing away. So they're stimulated. So up here, the pain receptors in your skin, the glass cut them, and so they're going to be stimulated. You'll feel pain. Yeah.
Weren't there also deeper receptors that were pressure receptors?
Yeah, the Pacinian. Yeah, the corpuscles. They're deep pressure receptors.
And wouldn't those respond to a lot of swelling in an area?
Yeah, they do. And then they're all, you know, that, and so do the pain receptors. Maybe the pain. That's a good point. Because maybe the pain receptor wasn't cut, but it's responding to that pressure on it because of the swelling, like you said. Yeah.
There was a case study where you asked us whether rubbing the wound would have been
Yeah.
helpful. What were we supposed to say to that?
Just rubbing it.
Yeah,
That
and
doesn't work.
so the case study asked that the guy rubbed the wound and it helped. Why, is that true that it actually helped, and why would that be?
Oh, I forgot about that question. Yeah.
No, because it breaks up the clot, right? Like the,
I think let me think about that.
I just spoke that he should.
But it helps in the healing or helps relieve the pain? What did it say?
I think it was about his personal
Just didn't help
experience.
in any way.
It was his own subjective experience
Yeah,
of pain.
maybe.
That's what you're saying? Yeah. That's good.
Maybe it was placebo.
I know when you lick a wound, you have lysozymes, so you have antibacterial enzymes in your saliva. When you lick your wound, that's good because you're putting those antibacterial enzymes to kill the bacteria that could be in your wound. But so it said just rubbing it.
I was like,
I feel like it would stop the clotting, like any kind of like scab or like
I mean,
clots that would form to stop, like that
To stop the white
friction-
blood cells.
Yeah, that friction would like break off that platelet.
Potential platelet plug. I mean, that's a good hypothesis just from what we've talked about.
Massaging his eye to reduce the scarring, but
I'll have to reread the question because, yeah, what's the endpoint you're looking for the rubbing?
Would it be like stimulating the blood?
That's my first guess is it would stimulate blood flow to the area, but I don't know. It gets complicated. Yeah.
That was my answer too, was that rubbing the wound would increase blood flow to the area by stimulating the area.
I think,
What it would also do is it would allow you to activate all the nerve endings in that general area, which can be distractionary if you kind of override the pain by like massaging it.
yeah, exactly. Like override the pain, meaning you're sort of numbing those receptors just by stimulating them so much.
And you're stimulating everything within the area, not just the ones that are going to throb with pain.
Yeah.
It's distractionary, I think.
Okay. Distraction therapy. I like that. Okay, so these are the signs of inflammation. Does anybody know the Latin words for these? So, anybody want to show off their Latin? So the four endpoints, these are the four endpoints of inflammation. Heat, redness, pain, and swelling. But sometimes in the clinic they might teach it to you, well in the future they might teach it to you as in the Latin. So, does everybody know a tumor is a swelling, right, of cells? So swelling and inflammation in Latin, that's tumor. Tumor. What's the Latin, or what's the Spanish word for red? What is it? Oh, that's kind of close. Well, it's rubor. For redness, Latin is rubor. So, tumor, rubor, calor for calorie. Calor is heat.
calor is hot.
Calor is hot in Spanish. Okay, so that's pretty close. And then pain is dolor. So they might teach you the four endpoints of inflammation in Latin or English. And then you do all this stuff in inflammation, all these cell biology moves, and the endpoint is to heal. So inflammation is a pain, but you do it to heal. And then I just show you here that you need to know for the exam, you need to know the four endpoints of inflammation. So you don't have to know all this flow chart. It's just kind of cool to look at. And then apparently I like the question on pus. Everybody's had a wound and seen pus before, I imagine. It's made of, what have you got in there? You've got dead neutrophils, macrophages, and other debris from damaged tissue, like the cells in the area that got damaged. And I'm going to jump ahead so we can finish immune 1.
Neutrophils, bacteria. It's bacteria.
We talked about complement last week, didn't we?
Kills bacteria.
I think we talked about complement proteins. Do you remember where complement proteins are made? The liver, good. And remember, complement proteins, we talked about this like slats in a barrel. The complement proteins insert themselves in the plasma membrane of the target cell. So you imagine this is plasma membrane of the bacterial cell. Complement proteins insert themselves to make a hole, or a pore in this case. And then what happens to this cell that got stuck with these complement proteins? What happens to it?
Got stuck with them.
Stuck with them in a sense. They were stuck in the plasma membranes, complement proteins.
Doesn't it make a hole that you can inject something into the cell that kills it?
Yes, you can, or just the hole itself will cause cell lysis.
Oh,
So
because
the
it,
cell will burst.
yeah.
Because the osmotic pressure is not the same.
Okay. It's like
But then you're right, you can also, once you've made the hole, you can dump granzyme in there. And then granzyme initiates what? Remember the natural born killer cells?
Yeah.
Natural born killer cells can, instead of complement, they stick perforins. The target cell, bacterial cell can lyse, but the natural born killer cell can also secrete granzyme into the target, and then what happens to the target cell? It does, but it dies a special death.
Oh. Yeah.
Starts with an A. Apoptosis, yes, apoptosis. Good. So that's the granzyme part. It can die an osmotic death, or it can die the granzyme apoptosis death.
That was the last lecture.
Yes, yes, exactly.
Perfect lecture.
Yeah. Just like in,
Do that
it's like in
tomorrow.
Goldfinger. James Bond, Pussy Galore, in the jet, they blow a hole, Goldfinger fires a shot, and then the jet loses pressure. Exactly. All right, I brought in a James Bond movie.
You can't really say people without like giggling.
I tried to.
Didn't quite get there.
Okay. So anyway, complement. So this is how you activate the complement proteins, and it's kind of complicated. You can activate these complement proteins with the classical pathway, which is the one we're going to focus on because that's antibodies do that. But the point is, you can go through a bunch of biochemistry, and at the end of that biochemistry, you're going to have these activated complement proteins. These activated complement proteins can do two things that I want you to know for the exam. One thing the complement proteins can do is they can opsonize pathogens. So you can coat a bacterial cell with these complement proteins. And what does that make the bacterial cell? What happens to the bacterial cell if it gets coated with complement proteins?
It looks yummy to
It looks
phagocytes.
yummy to what?
Phagocytes?
To the phagocytes, yeah. And then also complement proteins can do what I just showed you in the cartoon. Put these slats in a barrel, opens a hole into the bacterial cell, and you get osmotic lysis. Complement proteins do other things too, but I want you to know C3b. So complement protein 3b does these two things. That's what you should know for the exam. And what are the four endpoints of inflammation in Latin? Heat is calor.
Calor.
Heat is
Tumor.
calor. Swelling is tumor.
Redness is rubor.
Redness is rhubarb.
Pain is dolor.
Pain is dolor. Good. And I think that's the end of 1. Yeah, that's the end of 1. Any questions on Immune 1? That was the easy one.
Oh,
Now, hopefully you're warmed up for Immune 2.
What?
Which is the hard one. Do you guys remember where dendritic cells are found from 201? Remember back
In the brain.
That's a good guess because dendrites are structures on a neuronal cell body.
Yeah.
And that's tricky. I'm glad you said that.
The nervous system.
Well, no. Neurons
Uh-huh.
have projections called dendrites.
Uh-huh.
This is not that. This is, but it's the same word. This is a dendritic cell, not a projection of a neuron.
Oh.
But we did see a dendritic cell in 201. It has a specific location. Does anybody remember where they live?
In the body.
They do.
Yes.
Now help her out. She started you off. Now you guys help her get more specific. No one remembers from
Does
201?
that have another name for dendritic cell? Is it one of those?
Well, it is gonna, in this lecture, it'll be called an antigen
This
presenting cell.
assistive cell?
Yeah, an antigen presenting cell.
Would it be a
Yeah.
spinal stem?
No, where do you find it? In the cell, in the skin.
Oh.
Oh, is that Merkel?
It's in that same area where you find a Merkel disc.
Okay.
Yeah. Dendritic cells are found in the skin. That's 201 stuff. Now we're going to take a dendritic cell. It's living in the skin. If a bacteria gets into the dermis, the dendritic cell is going to find that bacteria, engulf it as a phagocyte, and then it's going to take that bacteria, go to a lymph node, and present the bacteria parts to B and T cells. That's antigen presentation. So the dendritic cell is a member of antigen presenting cells.
Okay.
Because what they do is they find bacteria and then they present bacterial antigens in the lymph node. Keep track of this guy. He's going to play a really important role in a super complex pathway. So,
God.
yeah. And then this is cool. This is atomic force microscopy. So it's an atomic force micrograph. Doesn't that sound super cool and new age kind of? I was gonna say new agey, but new agey means like when you have crystals and crap like that. So that's wrong. But this is an actual photograph of an antibody. Can you kind of see its Y shape?
Mm-hmm.
Mickey Mouse.
Kind of like this. Yeah.
It's like Mickey Mouse.
Let me see. Oh yeah, it does. It's a little mousy. All right, so let's check this out. The top of this cartoon, it says adaptive defenses. What's another name for adaptive? Specific immunity. Okay, so we've left non-specific innate immunity. Now we're in a specific or adaptive immunity, and it's all about B and T cells. There's other cells, but the major players in adaptive immunity are B and T. So here's a bone. What bone do you think this is, just from the cartoon? It's the femur. Yeah, look at that giant head on the femur. So here's the red bone marrow. Lymphocytes are being produced. What's the name of that process where you make white blood cells? It's one of those H words. Hemopoiesis. Yeah, good call. Hemopoiesis. You're doing it in the red bone marrow. You're making lymphocytes. And what are the two types of lymphocytes? B and T. B and T cells. The lymphocytes that stay in the, so they're born here. B and T lymphocytes are born in the red bone marrow. Don't be tricked on this test question. B and T lymphocytes are born in the red bone marrow, like you're born somewhere, but then you might go somewhere else to mature. B lymphocytes stay in the red bone marrow and become mature. T lymphocytes go to the thymus and mature. And then if you see the flow chart in the cartoon, once the T cells and the B cells are mature, they go into the blood and they find a lymph node. And they live there. Have you ever been sick and your lymph nodes get swollen?
Mm-hmm.
That's because whatever the infection is causes the B and T lymphocytes living in the lymph node to proliferate. So where you may have had 10 B cells, now you've got 10 million B cells in that lymph node, and the lymph node gets huge.
Is that the same thing that happens when you have like cancer?
Cancer of the lymph nodes?
Like my mom had
Stage
breast
4.
cancer and she had to have all her lymph nodes removed here.
Yeah, it's the same idea. It's a proliferation of cells, an uncontrolled proliferation of cells in cancer, but this is a controlled proliferation of cells.
Oh, okay.
And then it shows here in the lymph node, it shows what's going on in this green colored structure. Antigens, in this part of the cartoon, antigens are binding to a B cell receptor. So this little Y thing is a B cell receptor on a B cell, and it's binding antigens. And then when the B cell receptor binds the antigen, it's a foreign antigen, I should say, it's going to make a clone of B cells. And then what do the B cells make? What's the clone of B cells going to make for you? Antibodies. Yeah. And then those antibodies go into the capillary, and then the antibodies travel all over your bloodstream. Yeah.
So basically, the B and T cells are born in the red bone marrow. They move to their parents' house in thymus.
Yeah, that's good.
Grow up, and then they move out on their own into
Just the
lymph
T
node.
cells.
Oh,
Just the T cells go to the thymus. The
Okay.
B cells live in their parents' basement until they mature.
Okay. Gotcha.
That's good.
And the lymph node is the batch pad.
Yeah, the batch pad. Yeah, and
Hanging
they
out
go
with-
there.
Hanging out with their friend.
Well,
antigen?
actually, that's where they do all their work. So they're not having a lot of fun there.
Oh.
So I don't know if I'd call it. I'd say
The factory.
the mines. Yeah, the factory or the mines. That's where they go to do their work.
T cells or neutrophils?
Oh, the color is B. By the way, for this exam, the color is B for this PowerPoint.
They call them T cells.
The color is B. The color is B cells. Okay. And this is tricky because
Oh gosh.
the blue writing is part of the cartoon, not my blue.
Oh,
I
okay.
think her blue's like.
However, we will go through all of this in detail. So, I mean, either way. This kind of gives you an overview of what we're going to be doing. So you can use this to study too. Oh, we've seen this before. Yeah, here's the target cell. Here's natural born killer cell. Putting perforins. What's the other kind of protein you could use to puncture a hole in an enemy cell? No, the other kind of protein that you could use to put a hole.
Oh.
The other kind of perforin is a protein. This other protein you can stick in a cell to cause that osmotic lysis.
Complementary?
Complement proteins. Complement proteins. So two kinds of that'll definitely be on the exam. There's two kinds of proteins you can use to poke a hole in the cell. You could use perforins or you could use complement proteins. So in this cartoon, you've got the natural born killer cells using perforins. And then after that, after he's punctured a hole, if this cell doesn't die from osmotic lysis, the natural born killer cell is going to pump in granzyme, which is an enzyme that initiates apoptosis that will kill the enemy cell. And here's all this is testable. Looks like natural born killer cells. It's non-specific lymphocyte. Don't be confused. There's 3 lymphocytes that we're talking about: B lymphocytes, T lymphocytes, and natural born killer cells are lymphocyte. Natural born killer cells are not specific. B and T cells are specific. And then what does this mean? Use their TLRs to recognize non-self cells. What is the Toll-like receptor or the TLR on the natural born killer cell? What is that recognizing on the foreign cell?
MHC. No.
That's more of the specific stuff.
Yeah.
Remember, TLRs recognize on foreign invaders. PAMPs,
PAMPs.
pathogen-associated molecular patterns. TLRs recognize PAMPs on non-self cells. Non-self cells are bacteria, parasites, transplanted cells, all those are non-self. Oh, and I know what you were saying, because you saw non-self.
Yeah.
Yeah, non-self cells would have non-self MHC proteins, so.
Yeah.
And then there's a cell called a cytotoxic T cell, and I write it as T sub C. Cytotoxic T cell, it's a specific lymphocyte. Cytotoxic T cells use T cell receptor to recognize non-self cells. So there's T lymphocytes, we talked about those, right? T lymphocytes. And then you can break T lymphocytes down into cytotoxic T lymphocytes and helper T lymphocytes. So that C is a cytotoxic T. But, so in this cartoon, instead of an NK cell, you could call that a TC cell or a cytotoxic T cell. But instead of using a toll-like receptor, if this were a cytotoxic T cell, it would use a T cell receptor to recognize the enemy. The enemy is always a non-self cell, by the way. But whether you're a natural-born killer cell or a cytotoxic T cell, you kill the same way. You use perforins and granzymes and apoptosis. Oh my. That was fun. What? Yeah, thank you. Oh, I have some. That's inappropriate. What is it that they? What? Okay. Monkeys. Oh my. What are the three things that they say? Tigers and bears. Oh my. Because I have another 3 things that you should be afraid of. I'll tell you after. But it's pretty funny.
Yes.
Yeah, lions and tigers and bears, oh my. Perforins and granzymes and apoptosis, oh my. Oh my for the foreign invasive cells. So, actually, there's 3 types of there's 3 types of T lymphocytes. Cytotoxic T, helper T, and then we don't talk much about this, but I like this name. It's a regulatory T, so I call it a T-reg. Helps me remember, T-reg, yeah. And remember, so those are the T lymphocytes, and then we have B lymphocytes. They're the ones that make antibodies. We've kind of talked about antigens. Remember we first saw the A and B antigens in blood groups. Antigens are just, usually they're big proteins that can be recognized by antibodies for this legend. So here's the antigen, and you see its little shape. It's got a specific shape here that will bind to this blue thing, this antibody. But look at the antigen shape here. It will bind to antibody B shape, and then see antigen, this part of the antigen will bind antibody C. And so this whole thing is called the antigen, but these little pieces here that will bind the antibody, those are antigenic determinants, also known as epitopes. So now, for the rest of immune 2, if I say antigen, I mean antigen epitope, or if I say epitope, I mean antigen epitope. So antigen's just the big thing. And then the epitope is that little part of the antigen that the antibody recognizes. Yeah, it's a little shape that the antibody recognizes. Question?
Are they keyed like that always? Like is C always going to have the puzzle piece looking in? Or?
Well, I can say this. C, this shape on epitope C, let's say it's epitope C, will only be recognized by antibody C. Well, what are you saying? Will it always have the puzzle shape? I don't know.
That answers it. I didn't know if that was sometimes going to be A or B or C, depending on just whatever it was.
Yeah, so whatever shape this is, it can only be recognized by a very specific antibody. So for example, antibody B cannot recognize this part of the epitope. It can't because it doesn't bind to it. So antigens are complex molecules that can provoke or they can generate an immune response. And we saw that with blood grouping. The antigen A, you remember antigen A in the ABO blood groups? Antigen A was recognized by what antibody? A. Ha! Damn it.
Trick question.
It's really not that tricky, but people always get tricked by it. Antigen A on your red blood cell was recognized by antibody A. And in this case, when an antibody recognizes an antigen, you're going to get an immune response. Let's see. Antigen receptors on immune cells and free-floating antibodies in the blood can recognize the antigen. So, for example, this would be a free-floating antibody in the blood. It's an antibody, the blue thing. But this blue antibody could also be stuck to a B cell. And that, if this were stuck to a B cell, it would be called an antigen receptor on a B cell. And then we said this, antigens have epitopes. That means maybe self or non-self. So, self and non-self. Any protein on any cell of your body would be on a self cell, and your immune system shouldn't recognize it. But if a bacteria comes in and it's got non-self antigens on it, then your immune system cells will recognize non-self antigens on a non-self bacterial cell.
Okay.
So yeah, the immune people, the immunologists, they talk about self and non-self a lot. So non-self is always the foreign invader and will be attacked by your immune system. Yeah.
How does your body recognize the bad self, like the cancer cells? Do you have to, does an antigen presenter have to go?
When a good cell in your body becomes cancerous, that cancer cell now starts making proteins that look like non-self proteins to your immune system. So you hopefully, I mean, right now we're all developing cancer, but our immune system is going, oh, there's non-self antigens, let's destroy this cancer cell. So, this is a cartoon of the plasma membrane. I just put, you don't have to know all this down here. But when a bacterial cell comes into your body, here's its plasma membrane. It's got all these things sticking out of it. All these things are just antigens. Antigens, it's just anything that can be recognized by an antibody. Oh, here's my drawing. This will clear it up for you because my drawing is so amazing. So this green line is the PM. What's that?
Plasma membrane.
The plasma membrane. And then the yellow rectangle is the antigen, and then all these different shapes are the epitopes of the antigen. And it doesn't look like I'm going to test you on any of this. You'll probably get all this in micro. So if this could be the green plasma membrane of a bacterial cell, and any of these things can be recognized as an antigen. You know, does everybody know what a flagella is? So it's the little tail. Sperm have flagella, bacteria have flagella. They allow the bacteria to move. That proteins on the tail, the flagella, are antigens that can be recognized by our immune system. And these things are non-microbial antigens. Egg whites, probably some people know that you can't get a vaccination if that vaccine was made in an egg because you're allergic to egg whites. Or anybody have that? Know about that? Yeah. So that's just, there's some protein in the egg white that your body recognizes as a non-self antigen, and your body attacks it. And then you have an immune response that could go awry. Okay. And I just say antigens with their epitopes, that's the yellow box with all the little designs on it, will trigger the lymphocytes of specific immunity. So that's B and T cells. Different lymphocytes in your body will recognize specific epitopes. So all that means is if I'm a T cell and I have a T cell receptor on my plasma membrane, I have a T cell receptor that will only recognize the black dot, not the blue or the red shape. So right now, in this room, we have similar and non-similar B and T cells with their B and T cell receptor. So here's a B cell, and here's its B cell receptor. Here's a T cell, and here's its T cell receptor. In our population of people, some of our B cells are the same and some are different. That means if a foreign invader comes in, foreign bacteria comes in, some of our B cells will not recognize that foreign invader, and that foreign invader will kill us. But some of us, our B cells will recognize the foreign invader and be able to kill the foreign invader, and then those people will live. That's evolution by natural selection. So evolution designed this system where if a new antigen comes in, maybe, I don't know, from outer space or whatever, or a mutation, and you guys know this, things are mutating all the time, right? Some of us are going to live, some will die, like the bubonic plague. Some people lived through the plague and some people didn't. People that lived had B and T cell receptors that recognized Yersinia pestis, and then they could kill it. Am I creating enough drama here to engage you to learn more about the immune system by saying killing and death, stuff like that? Some of you will live, some of you will die. Have I piqued your curiosity? Okay, good. I'm going to work on my theater performance of that, but I'll get it. Specific immunity. Okay, do I want to talk about? Yeah, this isn't on the test. I'm going to skip over that. Immunogenicity and antigenicity. Because I didn't ask that. Let's do this and then we'll take a break. I've been talking about these guys, major histocompatibility complex antigens, or MHC proteins. And do you remember the analogy I used, the military analogy I used? Your MHC proteins on your cells are like what? Dog tags. Yeah. So this is a nucleated self-cell plasma membrane. All that means is this is a cell of your body. It could be a lung cell, a heart cell, a liver cell. It's got a nucleus. It's a self-cell, so it's in your body, and it's a plasma membrane. And then sticking out of that plasma membrane is this thing, your dog tags, set of dog tags called MHC class 1.
What does MHC stand for again?
Major histocompatibility complex. And all these different shapes, they're just different proteins. And then it's got this little area here called a peptide binding cleft. Do you remember what a peptide is?
Chain of amino acids.
It's a chain of amino acids. Yeah, it's just a short protein. It's called peptide. Wow, all of it came up at once. Dang, I don't want that. Oh well. Nucleated self-cells have self-MHC1 proteins. And what they do, these self-MHC1 proteins, they present pieces of intracellular self-proteins on the plasma membrane surface. So here's what's happening for this first dash. In all the cells of your body right now, you've got self-proteins inside the cell, right? So think about your favorite cell type. Is it a heart cell? Is it a brain cell? What's your favorite cell type? Inside that cell, there's proteins. And those proteins are constantly being chopped up, and the self proteins are being presented to the immune system on the outside of the cell. So if I'm a immune system cell and I come along and I recognize that there's a self protein, a self antigen here, am I going to destroy this cell? No. So that's happening all the time. These MHC class 1 molecules are presenting pieces of self proteins to the immune system. And the immune system comes by and goes, oh, and says, oh, that's a self protein. This cell's okay, don't destroy it. But what happens if this self cell got infected?
Maybe you won't be able to recognize other cells. Or...
Well, there is going to be a recognition process. So let's say this cell got infected. The same cell biology, the same process that chopped up intracellular self proteins is going to chop up intracellular non-self proteins. The same process is going to present that non-self protein out here in the peptide or protein binding cleft, and now that's a non-self protein. Now the immune system comes along and detects, oh, there's a non-self protein there. What's it going to do to this infected cell? It'll destroy it. So your immune system will destroy this infected cell, but your immune system knows to destroy it because there was a non-self protein in MHC class 1 binding cleft. See how it gets super complicated really fast? Yeah. Everybody's like, oh. You can also do this if this normal cell became a cancer cell. That cancer cell will now present abnormal cancer epitopes or abnormal cancer proteins in this binding cleft, and hopefully that cancer cell will be destroyed before it becomes a tumor. So all the cells of your body that have a nucleus are doing this all the time. They're always telling the immune system cells and the outside of the cell, the immune system cells, they're telling those cells we have a normal self protein or we have an abnormal non-self protein inside us. It has to be this way because the immune system cells, they can't get inside this cell. This cell, this nucleated self cell, has to show the immune system what it has on its inside. So, and something that might be a little confusing too, the epitopes, the antigen and epitopes are usually proteins. So, sometimes they use those terms all interchangeably. They're all the same thing. Protein, antigen, epitope can all be used interchangeably, which is confusing, I know. Have I given you enough time to smell that fine wine? Take a sip of it, swirl it around in your tongue, and okay. Well, then we'll take a break and come back in 10.
Whiskey, not wine.
Or that fine whiskey, whatever you like.